Welcome friends. Today I discuss about the different correlative aspects of water absorption in plant. Water absorption is highly regulated by transpiration in plant. Atmospheric gaseous status is also an important prerequisite for it. Physiological and pathological circumstances of plant also prevails water absorption. Here is a diagram that shows three important biological processes, namely transpiration, photosynthesis, and water absorption. These three processes are interconnected with each other. Welcome friends. Today I discuss about let us discuss about the relation between water absorption and transpiration. The ancestors of modern day plants evolved in water, and the internal environment of a plant has 80 to 90% water. The relative humidity inside plants is in between 95 to 98%. Land plants have evolved a number of mechanisms, for example, waxy cuticles, sunken stomata, etc., to ensure that they do not dry out and remain hydrated for their growth. Water absorption and transpiration are interrelated biological process. The water that is used in photosynthesis and subsequent growth is less than 2% of total plant water use, but the rest water evaporates out of the leaf as it captures carbon dioxide from the atmosphere. That means 0.035% carbon dioxide. This gas exchange occurs through stomata. Here, stomata is 1% of the leaf surface area. Here is a diagram that shows the relation between water absorption and transpiration. There is a relation between water absorption and transpiration. The relative amounts of photosynthesis and transpiration occurring at any one time depends on amounts of carbon dioxide and water in the atmosphere, inside and outside the leaf respectively. For example, flow of carbon dioxide and flow of water is calculated. Flow of carbon dioxide from outside, that means atmosphere, to plant cell is regulated by pressure difference, and in another way flow of water from plant cell to atmosphere is also regulated by pressure difference. So carbon dioxide enters into the plant cell and water exits from the plant cell. This shows that every unit of carbon dioxide used in photosynthesis, that plant loses about 66 units of water. This is known as transpiration ratio or water use efficiency, and varies between 10 and 100, depending on the environmental conditions. Here is a diagram that demonstrates transpiration and gaseous exchange from leaf and water absorption by plant root. To discuss about the correlative aspects of water absorption and transpiration. The plant can control the loss of water from its leaves by varying the aperture of its stomata. If a plant restricts the flow of water vapor out of its leaf, it automatically restricts the flow of carbon dioxide into the leaf for photosynthesis. This is known as transpiration compromise. For each of 100 liters of water used by a plant, the process usage is as follows. In case of photosynthesis, 0.1 liter water is used. In case of plant growth, 1.9 liter water is used. And in case of transpiration, the maximum water, that means 98 liter water is used. So continued hydration is essential for plant growth, but this is largely influenced by the control of transpiration. Here is a diagram that shows how water moves through a tree. There are some physical blockade in relation to water absorption. In the area of the path of water absorption, the physical blockade are: firstly, on the root hair. The root hair cannot absorb water mainly due to less density or due to some pathological condition caused by a mycelial fungi. Secondly, on the cortex. The cortex is an intermediate area between the epidermis and endodermis. It can be blocked mainly due to the development of physiologically inactive region in the symplastic pathway. Thirdly, on the endodermis. The endodermis is well protected by Casparian strips, which provides mechanical support to the plant roots, and the scarcity of passage cells in between reduces the amount of water taken up into the vascular strands. Let us discuss about the physical blockade in relation to water absorption. Fourthly, at the entry point of xylem, the penetration of water molecule through the dead wall of the xylem requires greater intensity of the root pressure above 2 atmospheres. But due to the reduction of root hair density, the pressure cannot develop and the water molecules fails to enter the xylem vessel. Fifthly, in the xylem element itself, the xylem vessel is a long channel connecting the root with the stem. It is blocked due to various reasons. The first reason is due to air embolism. Anytime the air bubble slipping into the xylem may cause air embolism and in the process reduce the upward translocation of water by causing air gap. Here is a diagram that shows normal xylem tube with normal water flow, and in another side embolized xylem tube that shows broken water column. There are some physical blockade in relation to water absorption. The second reason is due to formation of tyloses. The tyloses, which are ball-like projections in the inner wall of the xylem vessel, are formed due to some infection stress. They reduce the cavity of the xylem, so the area of water inflow is reduced. The third reason is due to fungal colonization. The fungal mycelia entering through the root colonize within the xylem vessel and cause blockage of the channel. And fourth reason is reduction in leaf density. The reduction in leaf density is seasonal due to abscission of leaves, as a result of which the transpiration pool is reduced and passive absorption is hampered. Here is a diagram that shows tyloses formation in xylem vessels. There are some physiological and pathological blockade in relation to water absorption. The cause of blockage of xylem or the water conduction pathway can be due to physiological or pathological reason. These are as follows. Under physiological reason, the first point is low density in capillary water. Due to extremely low density of capillary water, water availability to the root hair is extremely reduced. This is common in arid and semi-arid areas. The second reason is drying of the root hair. Due to extreme dryness of the soil particles, the root hairs get dried up and cannot absorb water molecule. The third reason is air blockage. The air bubble within the xylem vessel either reduces the water uptake or totally stops conduction, causing wilting. Another important reason is pathological reason. The infection of pathogenic organism can block the water absorption or conduction process by colonizing in the root or shoot xylem. These are as follows. The first reason is root knot nematode. The nematodes like Meliodogain species colonize in the cortical region of the roots of different crop plants of families like Solanaceae and produce hyperplastic disease like root knot and thus inhibits the water absorption. The second reason is root invading fungi. Fungal mycelia usually enter through the epiblemma but many of them colonize in the root itself, causing hyperplastic disease like gall infection. Here is a diagram that shows areas of blockage in the water conduction pathway and also depicts the leaf pathogen and root pathogen. The third important pathological reason is shoot colonizing fungi. The most common site of fungal infection is the shoot xylem vessel, which block the vessels and prevent water conduction through the xylem vessel. For example, wilt disease of plants. And fourth reason is leaf invading bacteria. These bacteria mostly invade the vascular tissues of the leaf, and as a result of which transpiration is inhibited and the passive absorption is impaired.